Thank you all for joining us. Thank you for joining us tonight. My name is Nancy Touhill. I am the peer facilitator for the Triple Negative Breast Cancer Let's Talk About It group. Before we dive in, I just want to go over a tiny bit of housekeeping and let you all know that we are going to start to expand the scope of the triple negative group, and we're going to make it into a breast cancer Let's Talk About It group and invite other members of the breast cancer community to join us. And then we are going to offer some additional triple negative specific programming through Share. So stay tuned for more details about that. You'll see some emails come out, but just wanted to personally let everybody know that as well.
Don't make it too easy.
I'm going to go ahead and mute anybody that's not on mute as well, just so that we can keep moving. Before we kick off, I'd like to tell you a little bit about Share. Share is a national nonprofit that supports, educates, and empowers anyone who's been diagnosed with women's cancers and provides outreach to the general public about signs and symptoms, because no one should have to face breast, ovarian, uterine, cervical, or metastatic breast cancer alone. For more information, please visit sharecancersupport.org. We ask that you all stay muted during this presentation tonight. When Dr. Schroeder finishes presenting, we'll have a Q&A part of the discussion. Please enter your questions into the chat, into the Zoom chat feature, and I will ask them on your behalf. We are recording this presentation, so anybody who speaks will show up on camera. So please enter your questions in the chat. This presentation will be recorded and available on the website in 1 to 2 weeks. So now I'm going to hand it over to our speaker to introduce herself. Dr. Schroeder, thanks so much for joining us tonight.
Thanks for having me. Good evening, everyone. I've been asked to tell you a little bit of background about myself before I do dive into the presentation I've made for you tonight. So I am a professor and department head at the University of Arizona out in Tucson. I've been here since 2002. I've been working in the field of breast cancer since I was in grad school, so in the mid-90s. And my lab here at the University of Arizona really tries to understand what are the drivers of triple-negative breast cancer, and then to also develop therapeutic interventions. And what I'm going to tell you about today is sort of an overview of triple-negative breast cancer, and then the focus that we have taken to develop some therapeutics. But before I get started, I just wanted to let you know that I'm a PhD, not an MD, so that means that I'm a scientist, not a clinician. I work with clinicians a lot. I have great friends at the University of Arizona Cancer Center, and I've worked in a cancer center since I was in graduate school. So I just want to make sure everybody understands that I'm not here under the auspices of being an MD who actually treats patients. Okay, so I'm going to go ahead and pull up my slides so we can get started. All righty, we good there? Okay, all right. So I'm starting off with a picture here in the background of normal versus metastatic breast cancer cells. So you can see the point of view from which I'm looking at this. So I'm going to tell you a little bit about different cancer therapeutics, which ones have worked, which ones haven't worked, and the direction that we're taking. And a lot of what I do in my lab is trying to really understand the difference between the normal cells, which we can see here on the right, and I'll talk a little bit about what you're actually seeing, and the metastatic breast cancer cells here on the left, because my goal isn't just to understand metastatic breast cancer, but I really want, and I believe it's possible, to develop a targeted therapy that only hits the cancer and doesn't hit the normal cell, to try and avoid all the terrible toxicities that we know are associated with therapeutics. So that is the goal of my research, to understand it and to find a way to target it and to avoid targeting normal cells while we're doing that. So a little bit of an overview of the cell types that we're looking at, just so we're all on the same page. What you're seeing in the slide is a cartoon on the left of the cells of the breast, which are actually pretty unique from most of the other cells in the body. So the cells of the breast form two layers And both of these layers can actually become transformed and turn into metastatic cancer. So in the cartoon, you can see a cross-section of a duct, and you can see little pink cells around the edge, and you can see some blue cells in the middle. And then in the very center called the lumen. So if we were to imagine this is a lactating breast, so it's producing milk, these blue cells would be producing milk, and the pink cells would be sort of the muscle cells that push the milk into that center lumen to feed the child. So this is the general structure of the breast. And when we think about breast cancer, we actually know that these two different types of cells seem to be driving the different subtypes of breast cancer that we know about. And this is the cartoon form. What you can see here in blue and green is what they actually look like. So these are some breast cells. This is just the luminal cells that we can grow in the lab to try and study how they grow and how they're different from the breast cancer cells. And so you'll see this picture a couple times. So I just want to outline what you're looking at. So the blue cells, the blue dots in the center, these are the nuclei of the cells. So this is the place where the genes are that are being transcribed and cause the cell to behave in the way that it's supposed to. And then the green outlining it is the cell membrane. So the outside of the cell that's communicating with the rest of the world. And in the normal epithelium, this is the normal cells that we were just talking about. You can see how they're just really lined up really nicely and organized. They're all touching each other in a very organized way. And they all are sort of facing this central lumen that I described earlier. And this is really indicative of normal cells in that they not only have their genes are not mutated, they're not misregulated, but they actually look structurally very organized and under control. And this is one of the things that we know is lost as we go through transformation and cancer and metastasis. And one of the things that we look at in the lab as a marker for changes that are happening, because we know that as we change the genes themselves, this structure will change as well. So those two different cell types that I just described to you respond to hormones that everyone on this call is familiar with hearing about, estrogen and progesterone, and they mostly regulate those inside cells, those luminal cells. And when we think about subtypes of breast cancer, we know that these guys are regulating those hormone positive breast cancers. Alternatively, those cells on the bottom and those pink cells in the cartoon, they don't tend to express these, and instead they tend to respond to different things. And these other proteins are called a HER family of proteins, and a lot of you will be familiar with HER2 because that's a subtype of breast cancer that a lot of people are familiar with, and I'll talk a little bit more about those. But the HER family tend to drive very aggressive metastatic cancer, and one of those family members is driving triple negative breast cancer. So we'll get back to those in a second, but just to sort of orient you to who's being expressed where and what they're doing. Okay. So these HER family of proteins are expressed in these cells, but what are they actually doing? What they're actually doing is they have an important normal job to do. So in normal cells as well as in cancer, unfortunately, their job is to sit in the cell membrane. So you can see in this cartoon, we have a cell membrane here, and we have this structure of, it says HER3 and HER2 here, but there's any of the HER genes will do this. They poke through the cell membrane. They receive signals from outside of the cell. So all cells are sitting in a whole environment of tissues, sending signals back and forth and deciding whether they should grow or move or stay put. All these things are communications between cells and their environment. And these HER receptors are one of the key communicators in breast cancer cells. So the way that they do that communication is they receive a signal from outside. Here it's labeled ligand, but we can think of this as a growth factor or anything that's going to cause the cell to change its behavior. And when they bind that signal outside the cell, they start sending off signals inside the cell. We call this signal transduction, and we're not going to go into the details of what's going on here, just to point out that this is one of the major facets that have been therapeutically targeted for these proteins and other cancers. And we'll talk about what's going on with breast cancer. But in general, they receive a signal, they activate all these things, we call this a signal cascade happening inside the cell. And the end result of that is you get things happening inside the nucleus, which is shown here in purple, that change the different genes that are being expressed. So it changes the different proteins that are being made, changes the decisions that these cells are taking. So the biggest decisions that we think about for cancer is it helps the cells decide whether they're going to grow and divide, whether they're going to migrate and invade and metastasize. All those sorts of decisions can be driven by these processes. So just one more time, the different subtypes of breast cancer that everybody's heard of. So we have estrogen receptor positive cancer, which has lots of different names, but these are the sorts of cancers that people be taking tamoxifen or letrozole or something like that for. Then we have HER2 positive cancer, for which we actually have a really great drug, which is most commonly known as Herceptin. And then finally, we have triple negative, and it's called triple negative because it's not expressing the HER2 receptor, the estrogen receptor, or the progesterone receptor. That's why it's called triple negative. And unfortunately, that means we can't use any of these great therapies that we have for HER2 positive or luminal cancers because it's not making those proteins. And the hunt has been on for a long time to try and figure out what is driving triple negative, because certainly there's something driving it. And one of the key candidates for what's driving triple negative is HER1. And HER1 is the protein that I've been working on in my lab. Well, I've been working on HER1 since I was a graduate student in the middle of the 90s. HER3 is another family member. I'm not going to tell you much about that. It's a little bit different, and we'll just leave it to the side for now. But we're going to focus on HER1 as the driver of triple negative and what we've been trying to therapeutically target in my lab. So an overview of all these cancer types before we really drive into triple negative breast cancer, just to sort of help everybody put it in context of all the variety of information they have in their heads about all these different cancer subtypes. So we tend to think of, so we've got luminal A and luminal B. We tend to think of these as just a group of cancers, as the cancers that express estrogen receptor. And they're by far the most common. But they also tend to be diagnosed in a low grade. They tend to be diagnosed before they metastasize. Now, this doesn't mean they can't turn into really scary cancers, but because they tend to be diagnosed early and because we have these really, really effective drugs for them, the prognosis for them upon diagnosis tends to be a little bit better than it is for the HER expressing cancers. Now, both triple negative and HER2 tend to be diagnosed when they're high grade. They tend to have a poor prognosis because they tend to be diagnosed and already be invaded or metastasized. Now, for HER2, we have Herceptin, which is shown here with the drug company name trastuzumab, but it's Herceptin. And in about half of those cancer patients, it works pretty well. But when we get over here to triple negative, which is about 20% of all breast cancer patients, which is a big number, I teach classes on cancer biology, and we talk about all the different cancer types and the prevalence of all the different cancer types. And we know that the most common cancers, at least in the United States, are colon, lung, prostate, and breast. Breast is among women the most common right after lung cancer. And so even though we're only 20% of that number, it's a big number. It's about 40 to 60,000 women being diagnosed. Now, when we talk about responses, we actually know that of all these cancer subtypes, triple negative responds the best to chemotherapy initially. And this is actually because it's this high-grade cancer. So chemotherapy works by killing any cell that's dividing rapidly. And so this is why we have all the toxicities associated with chemotherapy, GI problems, anemia problems, hair loss problems, because these are cells in your body that are also dividing rapidly. And chemotherapy is not specific for cancer. It's just specific for cells that are dividing rapidly. And so this is why triple negative tends to respond well to chemo right off the bat. The problem, as everybody knows, is that resistance arises. And frequently when you have advanced metastases, they fail to respond. They fail to continue to respond to chemo. So we really need to figure out what's going on with these cancers and find some targeted therapeutic that's going to impact them. I think I've already said this, but basically it's just reiterating that when we're thinking about the HER-specific cancers, these are cancers that tend to be metastatic. They tend to be a lot of times metastatic when they're first diagnosed. And we think about metastases that happen in these patients. They tend to happen in bone, brain, lung, liver. And these are of course, just devastating diseases to have. So again, finding the reason why these HER-expressing cancers are growing and metastasizing is so important. So I've shown you a lot of cartoons up till now, but I also wanted to show you what the cells actually look like. So what I'm showing you here is an ultra high magnification of a metastatic triple negative breast cancer cell. And this is outlined here. And what it's sitting on is this sort of stringy things in the background. It's sitting on some artificial tissue. So it's got components of the same tissues that we have in our body that they would normally be grabbing onto. And one of the things that really strikes you as you look at these images of what these cells actually look like is their ability to reach out and grab the tissue that they're sitting on. Metastatic triple negative breast cancer is unfortunately really good at moving through tissues, invading through tissues, and then moving on to other sites. So as opposed to all the cartoons that we've been seeing up until now, this is what those cells actually look like. And so these are some of the cell types that we work with in the lab when we're trying to figure out what's going on in there, and then we treat them with therapy. And I can tell you, very satisfying when we find a therapy that we can kill these with. Okay. So let's take a view of what the current therapies are for these proteins. So this is another cartoon. It's listed as EGFR and ERBB2. This is just another name for HER1 and HER2. So the same proteins we've been talking about all along. And it's just showing them in a different cartoon. But this is them sitting outside of the cell. This is the cell membrane. This is the inside of the cell. And what we can see is that there's two different types of drugs which are on the market right now and work in a significant number of cases of non-breast cancer, unfortunately. So we have something called an antibody, which binds the outside of the receptor. And for HER1, this antibody works really well in colon cancer, head and neck cancers. In fact, in colon cancer, it's the primary therapy that people receive is this antibody against HER1, and it works really well. Unfortunately, doesn't work in breast cancer. And I'm going to tell you why I think that is, but it doesn't. Then we have a similar sort of situation, an antibody against HER2, and this is, again, Herceptin, works great in breast cancer. In fact, it's the poster child for this type of therapy because it works so well. The other way that we have designed therapies to hit these proteins are something called tyrosine kinase inhibitors. The inside of these proteins are enzymes, and those enzymes start that signal transduction process. So being an enzyme means that they can change proteins that they interact with. And so therapeutics have been developed that block the activity of that enzyme. So basically kills it and makes it a dead enzyme. And again, in certain cancers, these therapies work really well. So the TKIs against HER1 work really well in mutated cases of EGFR. So this would be much more commonly found in lung cancer. So a lot of lung cancers in people who smoked, the type of lung cancer that they develop has a lot of mutations. And this therapy works really well. You may have heard of people taking a pill and their tumors going away. That's this therapy. Unfortunately, it has no activity in breast cancer. So, and again, similarly for HER2, there's something similar. So by now, I think you've all got this this part of the story that these proteins are really important. We know they're driving cancer, not just breast cancer, but other types of cancer. We've developed therapies against them, but they don't work in breast cancer. At least the ones to HER1 don't. And so the question has been for a long time, why? What is going on different in triple negative breast cancer? We know it's making lots of HER1. We know how HER1 works. We have these therapies that work in other cancers, but it's not working here. So one of the things that I've discovered in my lab, but also other people around the country and around the world have figured out about HER1 is that in cancer, it behaves very differently. So I told you that it sits on the surface, receives a signal, and then sort of sends off signals on its way. What we know is in cancer is that it's not just sitting at the cell surface. So it's not up here in green like it would be with normal cells. Instead, it gets moved into the nucleus. And this is a process by which it binds its protein on the cell surface that it needs to interact with the outside world. And then it grabs it, and instead of doing what it's supposed to, it actually heads inside via some really novel routes, goes right into the nucleus itself, and changes all the expression of all the genes itself directly in the nucleus. And when everyone discovered this about 15 to 20 years ago, everyone was shocked because these kind of proteins shouldn't do that. But the reality is people had been studying them in normal cells so long, or in other cancer subtypes so long, they never had seen them go into the nucleus. It was only until we started really looking in certain subtypes, specifically triple negative breast cancer, that we found them doing this very strange thing. And the interesting thing about them doing this very strange thing is that could easily explain why they're not responding to those other therapies, because those other therapies require them to be sitting on the cell surface and then to be activating that signal transduction. And they're not doing that. They're going all the way into the nucleus and not activating signal transduction. So the hypothesis in my lab for a long time has been, well, if we can figure out what's taking them into the nucleus, could we develop a drug to block that and keep them from going into the nucleus? And so the therapy that I'm going to tell you about today is the result of a couple decades of trying to understand this process and develop drugs. So I'm going to tell you about one drug that we've developed that's working really well, but it's not the first one that we ever tried to develop. It's actually the third one that we've tried to develop. And the other two, even though they looked really promising at early stages, they fell apart for different reasons. This one is the first one that's made it as far as it's made, and I'll show you where we're at. Okay, so everything from here on is going to be talking about this nuclear HER1. So the one that's not sitting up here on the surface, but instead is down here inside the nucleus directly doing things that we don't want it to be doing. So this is a very, very busy slide, but really it's to point out the differences between these two pathways. So if we look at this dotted line in the middle, everything on the right is what we used to know about what HER1 does, all that intermediate signal transduction, binding ligand, activating those enzymes. That's all the stuff that we used to know. Everything to the left of the dotted line is this new pathway where it used to be sitting at the surface and it gets traversed all the way in through the cell to where it's inside the nucleus itself, driving these gene transcription. And we know that it can drive all these things that we associate with cancer progression, allowing the cells to grow, survive, invade. And then I'm going to tell you a little bit how it actually modulates the immune system as well. So here's another very busy cartoon, but again, let's just focus on one little part of it, and that is the part of it going to the nucleus. If we look on the right-hand side, here it is sitting at the cell surface. All proteins that sit at the cell surface go through a process where it gets invaginated inside the cell. That's very normal. What's unique here is they start associating with a group of proteins called sorting nexins. And sorting nexins do what their name might make you think that they do. They sort things out. They sort things and send them to the right place. And these sorting nexins are the proteins that take HER1 into the nucleus. And what I'm telling you is the result of about 10 years and 5 different graduate students' PhD thesis. But that's what's happening. We have found the carrier that's grabbing EGFR and take it into the nucleus. Now, once we discovered that it was these sorting nexins that were doing the job, that's when we decided to go after them and try and develop a therapy, because we reasoned that if we could block the interaction between HER1 and these sorting nexins, and we couldn't have HER1 in the nucleus, then maybe that would be the thing that we needed to do. So the drug that we developed, and I apologize for this, is called SNIX 1.3. It's called that just because whenever we were developing drugs in the lab, we have lots of iterations and lots of changes, and we try and keep all those names in the name as we go forward. But that's what you're going to see pop up on all these slides is this name. Just going to call it SNIX 1.3. But whenever I'm talking about that, I'm talking about that drug that prevents HER1 from going into the nucleus by preventing it from interaction with those sorting nexins. Okay, so here's a couple of different pieces of data to show you that this drug works, and it works in human cells, works in human triple-negative breast cancer cells, doesn't work in normal cells, and works in a mouse model of breast cancer. So if we look at these graphs up on top, these are data in which we take human cells, we grow them in the lab, we treat them with the drug, and then we find out if they've survived or not. Okay, so if we go across the top here, all of the cells that have gone across the top, these are all surviving cells. Okay, and as they drop down to the bottom, these are cells that are dying. And one of the things that we do in all of our studies is we compare our drug, the SNIX 1.3, to the established drugs that people have already made against HER1. So in this example, we're comparing it against a drug called sunitinib. And what sunitinib is one of those drugs that blocks the enzyme activity of HER1. This is a drug that works really well in those mutated lung cancer samples that I was telling you about. We also know that it has toxicity. So it's a good way for us to compare how are we doing against the standard of care for trying to target this drug, target this protein, and then how are we doing for possible off-target effects, meaning are we going to hurt normal cells as well? And this graph on the left is triple-negative breast cancer cells. And we can see that when we treat with SNIX 1.3, we get great killing of these cells, rapidly killing them, and they don't come back. This is compared to that enzyme sunitinib that people use for lung cancer, which also works, but doesn't work quite as well. And then if we come over here to normal breast epithelial cells, so these are cells that are not cancer, we found something really exciting. And you know, I am super excited that this drug works against breast cancer, but I'm even more excited that it doesn't work against normal cells. And that's what this graph shows. So this graph shows that if we have these normal cells and we treat them with that enzymatic TKI, the one that works against lung cancer, it kills them. It kills them even better than it killed the cancer cells. This is one of the reasons we're seeing toxicity with these sorts of drugs, because they're not specific to something happening in the cancer, because these proteins are expressed on normal cells as well. But when we treated it with SNIX 1.3, it didn't do anything, which was really an exciting discovery. And when we really think about the mechanism of how this is working, we would expect it not to do anything, because in normal cells, EGFR or HER1 sits at the cell surface and hits off that signal transduction cascade, but it doesn't go to the nucleus. It's only in the breast cancer cells that we've seen that it goes to the nucleus. So that actually makes sense then that it's not impacting normal cells, because normal cells aren't doing anything. So SNIX 1.3 is going in there and it doesn't have anything to target, so it's not hitting anything. Whereas sunitinib is completely destroying the normal cells. So this was super exciting to us. We repeated it in a lot of other cell lines, but we really wanted to get it into an animal model. And so the data from the animal model shown here on the bottom. And the reason we need to do this in an animal model is that we frequently, I think, as a public, we think of cancer as there's the tumor, there's the cancerous cells, and they're behaving all by themselves. But the reality is there's no cell in the body that behaves all by itself. All cells in the body communicate with their environment. And that's the same for cancer as well. Cancer cells also have to communicate with their environment. And so we need to make sure that the therapies we develop will be functional when a cell is in the very complicated environment of the tissue. So it's not just the local environment, but it's all the hormones that are produced in an animal. It's the blood vessels, the immune system, all these things impact how tumors grow and how they respond to therapies. And so it's really important to test these in an animal model that has all those different things impacting what's going on with the tumor. So the animal model that we chose is an animal model of breast cancer that's driven by HER1. And you can see here on the left, this is the amount that the tumor has grown. And this is each individual tumors over the course of a study. We just sort of lined them up all here together. And this is a control animal, so they're not being treated with SNIX. Whereas when we treated them with SNIX 1.3, we had 2 tumors grow, but the rest of them shrunk. They shrunk down to the point where we couldn't find half of them. They were just gone. And this was just so exciting to us that we're not just causing stasis of the tumors, but we're actually causing regression of the tumors. So this is fantastic, letting us know that, that this pathway not only is important, but that we can develop a therapy to induce regression. And, oh, and I almost forgot to point out, so this study and the development of this drug was the work of a graduate student, Ben Outwell, who's still in the lab right now working as a postdoc. I wanted to, so something that we're, of course, extremely interested in is the toxicity. So I showed you some experiments where we found out that our drug wasn't doing anything to normal breast cancer cells, but that was just, or not normal breast cancer cells, sorry, normal breast cells, but that was just one single cell taken outside of the animal. So we wanted to go back into the animal and see if any of the tissues were being impacted. And so we treated mice 3 times a week for a month, and then took their tissues and looked for any sort of evidence of toxicity. We have veterinarians on staff here at University of Arizona who did that work for us. And they didn't find anything, no evidence of toxicity at all. So based on all that work that we found this novel way to target it, and we developed this drug. It looks like it's non-toxic, and it looks like it works really great in our mouse model. I'm working with a clinician here at the Cancer Center to try and complete any other requirements the FDA has so that we can apply for a clinical trial to try and get this in people and hopefully make a difference. Okay. So this is just a summary model showing all the things that I just told you about. But when we think about what's going on, this activity with the natural killer cells is really exciting, because we know from a lot of other types of therapies that people are working on that if you can activate the immune system against cancer, it really does an outstanding job of going against and not just killing the cancer in the breast, but the metastatic disease. And in fact, natural killer cells, that's sort of their claim to fame, is that they're really good at killing metastatic cancer. So that's one more reason why we're just super excited that this has a strong potential, and I can't wait to see what's going to do when we get it inside people. Okay, so that is all that I had to tell you about. I wanted to make sure that I mentioned Ben and Angelica, which I did. And these are all the other students in the lab working on this process. So I'm going to stop now and take questions.
Thank you so much. I mean, I didn't expect to have this reaction, but I found myself like tearing up a little bit watching the video of the cancer cells dying. That's incredible. And, you know, I think I can speak for all of us when I say thank you so much for doing that work.
Let me just tell you, we tear up too. We get that sort of results. We get pretty excited.
Yeah, it gives us a lot of hope. It does. And so we've had a couple of questions. So One is you mentioned HER1 being on the surface of the cell and also being in the nucleus. Is that, I wasn't clear, is it always on the surface and in the nucleus, or does it only go to the nucleus when it becomes metastatic? I wasn't quite sure on that particular one.
So it's always on the cell. It's always on the cell surface. And it's always on the cell surface in normal cells. So almost every cell in your body has HER1, which is one of the reasons why there's toxicity associated with that. It works at a very low level most of the time. But then in triple negative breast cancer, that's when we're seeing it on the surface and in the nucleus. And it's in there in the metastatic cells and in the non-metastatic cells.
Got it. Okay.
We actually know the reason, but it's sort of wonky to get into it. So I can get into it if you want.
No, fair enough. Fair enough. So you mentioned cancer invading and cancer metastasizing. Can you clarify the difference between those two terms?
So most of you have probably know about stage 1, 2, and 3, and 4 cancer, right? And so for any cancer to be called cancer, it has to have local invasion. So it can't still be sitting where it's supposed to be. And so this is why the first type of cancer that's diagnosed is stage 1. It's invaded, but it's still just right there at the local spot. If it hasn't invaded, this is where you have things like ductal carcinoma in situ, where some things have happened, but it hasn't invaded at all. So it's not technically cancer yet. And then the different stages go forward, sort of drawing it away from that first site where the cancer was formed. So stage 4 will be called a distant metastasis, and that means it's left the first site and now it's gone to bone, brain, lung, that sort of thing.
Got it. Can you speak to the clinical trial process a little bit more and how you would go about getting your drug approved?
Yeah, so it's very involved, but I try to remind myself that it's very involved for a good reason, because it's there to protect us, right? So the clinical trial process was developed when, in the 50s, physicians were just throwing things in patients that sounded reasonable, and a whole lot of bad things happened, right? So clinical trial process was developed to try and protect us. And so there's a whole lot of stages that you have to go through to get something approved by the FDA. So the two big ones are, the first is to evaluate toxicity, which we've done, and that part actually is ready to go. The second part is you have to find a way to be able to detect the drug in a patient after you've given it to them. And so the reason you need to do this is you need to be able to figure out, how much drug can I give this patient before it becomes toxic? How much drug can I give this patient and it's still circulating in their blood system? Like, how do we design the trial? How often do we give it? How much do we give it? And to do that, you have to have a method to detect it in blood after you've gotten it out. And that's where we're stuck right now. So we're working with a lot of different scientists who are very talented at these sorts of things. And we're having difficulty reliably detecting it. We can find it, but we can't always find it. And it's quite a head scratcher. And we don't know why, but we're working hard to figure it out. And so once we get those two experimental things done, the clinician has to write up a clinical trial that goes in with the application. And then we have to work with the companies who are going to make the drug, who are going to decide, how much drug to put in a bottle, how to make the label so that everybody's absolutely clear on how much of the drug to give a patient at what time. That has to be written out, really carefully. And so those all are processes that have to go through as well. And then we sort of take our whole data packet and give it to the FDA, and they look at it and decide if it's they think it's safe enough to try in a phase 1. So phase 1 clinical trial is just safety in a human being, because no matter how safe it is in a mouse, it's not a human being. It's just a guideline. And so a phase 1 is safety in a human being. And then if we pass that, then we can apply for a phase 2, which is where we can start looking at efficacy. Does it actually work on the cancer inside a person?
Got it. So I think we all can look back at our pathology reports and see, our HER2 designation or ER, PR. is there any testing for HER1 in pathology at this point, or is it just assumed that every cell has HER1? I'm curious about that.
No, there's not. And so the pathology that we're doing for, let's say, for example, for HER2, I think that's the best example. That actually took a really long time to get to the stage where pathologists are routinely looking for that. And part of that process was there's two different ways that it's evaluated. One is called IHC, where they look for the protein and they say, is there a whole lot of this protein around the cell membrane? And if it is, then they'll give you a grade 1, 2, 3. And then the other way they do is they say, is there been genetic alterations to the HER2 gene? And that's something called a FISH. And they'll tell you whether or not that is. And then they'll put those pieces of data together and they'll tell you, you're HER2 positive, you're HER2 negative. So for EGFR, or sorry, for HER1, we have a FISH test for it, for amplification, but it's much trickier than HER2 because it doesn't just stay at the surface, because it goes inside. It's hard for pathologists to give that a number. And then because of the reasons for driving it inside to the nucleus, that changes whether or not the FISH is important or not. So it's trickier than HER2, and we'll work it out. We'll figure it out eventually, but it's going to take some time to figure out all the details.
Yeah. Okay. And you just mentioned grades. Can you speak to stage versus grade a little bit? I don't know that we all were given, grade versus stage. We might have been one or the other.
Yeah. So not all pathology reports will have both. And I'm actually not sure why that's the case, because they should all have the same information. But regardless, grade is how much it's metastasized, how many lymph nodes are involved, that sort of thing. Stage. Grade is the cells themselves, if they are highly replicating. So if they see a lot of evidence of cells replicating, then that'll mean it's aggressive and it's not well differentiated, and that will give it a higher grade.
And there's 4 stages, 3 grades. Is that right?
There's definitely 4 stages. The grades change a lot depending on the subtype of cancer that you have. And even like different cancers, like melanoma has something like 12 grades. It's something that's really unique to the subset of pathologists who have decided to define what the stages and grades are for the cancer. So I can't promise that the grades are only 3 for TNBC. I don't know that that's true, but it might be true.
Okay.
This is one of those times it would be good for you to have a clinician here instead of us.
So you mentioned that HER1 can be found in other types of cancer, like colon cancer, and it's more treatable in those types of cancers. What other types of cancers might you see that particular mutation?
Probably prostate cancer. So prostate cancer, turns out prostate and breast have very similar structures. So that bilayered epithelium that I described at the beginning, they're not exactly the same, but they're the same. They're both driven by hormones, and they both express the HER receptors. So it's not as much work has been done there, but it's likely that's going to be the case. There's also a form of HER1 that drives glioblastoma, where this seems to be happening as well. So one of the drugs that we developed along the way getting to this one actually worked pretty well in glioblastoma models. So it's likely that process worked in there as well.
Wow, interesting. Do you think that there's a genetic link then to a HER1 mutation?
HER1 mutations, no one has found any heritable HER1 mutations. They seem to all be driven by environmental things like carcinogens. And the HER1 in breast is not mutated. It's just the normal one. It's just in the wrong place and it's overactive.
Interesting. Okay. Is there any hope or thought that the work that you're doing would be kind of adapted for the prevention of cancer instead of treatment?
Not that I know of. You know, when thinking about the prevention of breast cancer, that's going to be a tough nut to crack because most breast cancer is driven by hormones, and we're not going to get rid of our hormones. Hormones are good for us. We like them. And so to separate that from breast cancer, that's going to be tough. And I don't have any data saying that it's preventative.
Mm-hmm.
I also, the other thing about, I'd hate to start doing any treatments with HER1 inhibitors just because so many cells and so many types of tissues in your body need these for normal function. It seems safer for it to be a short-term thing than a long-term thing.
Yeah, I was going to ask, what good does HER1 do in the body?
It does everything. So the original name for this is called epidermal growth factor receptor, and it was originally discovered in that it made the hair and your skin grow, your teeth erupt, your eyes open. And when people started really digging into it, and they did things like they created animal models that didn't have any, the animals can't survive. So all of the organ systems in your body need it to function. And it's because it does this very basic function of allowing cells to grow, telling cells when they need to die, telling cells to migrate. And you can imagine, like, as an organism grows, like as a baby grows and you're forming all these tissues in your body, you need those sort of signals to happen. And HER1 is one of the major drivers of those signals.
So we all know that triple negative tends to recur perhaps more than some other subtypes of breast cancer. How does, can you talk a little bit more about that? How does the HER1 protein maybe affect recurrence, or are there any treatments out there that can help prevent recurrence, or anything on the horizon?
Yeah, so I mean, you guys know about the chemotherapy that people give when they have recurrent, and they'll switch you to a different chemo and a different chemo to try and find one that you're not resistant to. And unfortunately, this is why we still have 40,000 women a year dying from breast cancer, because this happens and we can't find a way to treat it. So HER1 plays an important role in metastatic recurrence of estrogen receptor positive disease, actually. So patients who are originally diagnosed with the luminal cancers and are treated successfully and will go for 10 to 20 years, and their cancer will recur, many of those cancers have suddenly turned on HER1. And so the idea is, well, let's give them one of these therapies that work in these other cancers so great, and then they don't work at all.
Yeah. Okay. You talked about natural killer cells in the body. Is there a way to boost those naturally to kind of help keep that immune response boosted?
There is a way to boost them. So there's proteins that are released in your body that cause them to grow and cause them to be more active. The problem is getting them to recognize the cancer, because the cancers can turn off all the lights, and even though the NK cells are maximally activated, they'll just cruise right by them. They won't see them. So the trick is to turn those on, which looks like we found a way to do it.
So would you put that in the kind of the immunotherapy category then, if they're revealing those cells?
Yeah, that's what we're doing. So we've got a lot of experiments designed that we're doing right now that I'm applying for funding for to really figure out exactly how it's doing it and see if we can amplify that in any way. That data, that's why I had you not record that little bit of data, because that's brand new data, and it's exciting, and Angelica's just really working on the early stages of it.
Yeah, that's awesome. Can you speak to any of the other immunotherapies then that are out there in the market for triple negative, like Keytruda? Does it work similarly to the drug in your lab?
So Keytruda is going to work against a different subtype of immune cells called T cells, T killer cells. And they have a different set of side effects that the natural killer cells will have. They have it's a different way to activate them than natural killer cells. Those drugs, and a study just came out for triple negative breast cancer that it is effective. It's just they tend to only work in about 10 to 20% of the patients, and it's because of the way that they work. So the way those types of immune therapies have to work is when a tumor is growing, these cells in your body called T cells will recognize the tumor, and they'll all sort of charge in and try and start killing it. But then they all get turned off. And the reason they get turned off is because they recognize the tumor as coming from yourself. And T cells are a type of cells that they really care about. Is this something not me, or is this me? And I'm not going to kill anything that's me. And so as the T cells run in there, they recognize that the cancer came from me. And so they stop and they sit there. And the drugs, the immunotherapies that are being used right now that have been clinically approved, they basically take that stop sign and they shut it off again and reactivate them. And one of the side effects is that the immune system can start attacking other parts of your body as well, because you've sort of turned off everybody's stop signs. So it's got some toxicity, but a lot of those toxicities can be taken care of with like steroids by your physician and hospitalization. The other thing is unless those T cells see a lot of mutations, they're not really going to attack in the first place. And breast cancer isn't a cancer that tends to have a lot of mutations. It's not driven by exposure to carcinogens or sun or any of those things. It's really driven by hormones, which aren't carcinogens, and they don't induce a lot of mutations. So because of that, it's always going to be a smaller number of patients who can respond to those immunotherapies. And this is also one of the reasons why we're super excited by natural killer cells, because they're agnostic to mutations. If they see something that they think should be killed, then they're going to kill it.
Do you envision this drug as a treatment for metastatic triple negative breast cancer, or would it be used as well in early stage cases in a perfect world?
In a perfect world, it would work for everything, but I don't have the data to support that that's true. So one of the studies that we are still working on is the metastasis study. And we didn't get that data at the same time that we got the first data, just because those animal models are different. We know in our experiments in the lab that it blocks experimental metastasis in the lab, but I haven't yet been able to test it in an animal model. And we just don't know yet.
Yeah, got it. How, in your mind, how far out are you from seeing this used in humans?
Oh yeah, I wish I knew. Until we get this experiment to work where we can detect it when we inject it, we can't put our application into the FDA. Mm-hmm. And experiments are tricky things to predict. It could work tomorrow. I mean, the data could be sitting there right now in my lab. I don't think it is, but it could be. And then again, it could be months, a year. I just don't know. And then once we get the packet into the FDA, how long is it going to take them to process it? I don't know. From the physicians that I'm working with at the cancer center, they think it should take about 6 months before they would get approval. And then the clinical trial has to be approved at the clinical trial sites, and that usually takes 4 months. So I don't know. I wish I knew.
Yeah. Hopefully you would get a phone call if that data was sitting in your lab right now.
Yeah. Everybody in my lab text me all good and bad. They text me.
Yeah, I would certainly hope so. Okay, one final question, and then we're going to have to wrap up. So we've heard about blood tests to detect the effectiveness of chemo, particular types of chemo. Can you speak to that a little bit? How important, effective are those blood tests? Would you recommend seeking those out just to kind of keep everything efficient and effective?
Yeah, I don't actually know. I don't know about the data. So you do, when you have cancer, tumor cells do circulate in the blood. And there's been a lot of work to evaluate pulling them down and using them as some sort of a way to discern how well you're doing. But I don't know about any of the data that's come out of there.
Yeah, got it. Okay. Well, we are right on time. Thank you again so much for not only taking the time to speak with us, but for the incredibly important and meaningful work that you and your team are doing. We are so grateful, and hope that you'll come back and talk to us again. We want to keep track of your work, so I'll keep an eye out for sure. I'm sure everybody else will too.
All right, awesome. Thank you. It was great being with you guys today.
Yeah, you too. Thanks again.
Bye.
Bye.